Assalamualaikum and hello everyone. Today our group will be presenting on the topic chromatography, candy dye separation using paper chromatography. This experiment focuses on separating dyes found in candies, which are actually quite similar to food dyes. That's why we chose to use food dye in our experiment as a representative sample. Before we begin, let me first introduce our group members. They are Puteri Damia Zulfah binti MARZUKI, Nur IZZATUL FAQIHAH binti JAMALUDDin, Muhammad Danial HAFIZI bin Muhammad RAZAK, NADHIRAH binti HARUN, QISTINA MARDHIYYAH binti MAHRUS, and lastly, Wan Nur ATHIRA binti Wan Muhammad RAZZI. I will introduce you to chromatography. What is chromatography? According to Premref and Zubair, chromatography is an analytical technique used to separate the components of a given mixture. This method is widely applied in both research and industrial settings. Now, let's look at the basic principles. In chromatography, molecules in a mixture are applied onto a surface or into a solid. These molecules then move with the help of a mobile phase while interacting with a stationary phase. The differences in how each component moves allow them to be separated from one another. Next, importance and types of chromatography. Chromatography plays a crucial role in many fields. It helps scientists to identify, analyze, and purify substances from complex mixtures. This technique is extensively used in chemistry, biology, forensic science, food testing, and environmental analysis. There are several types of chromatography, each suited to different applications. The main types include gas chromatography or GC, liquid chromatography or LC, thin layer chromatography or TLC, and paper chromatography. Each method operates on the same basic principle but varies in terms of materials and techniques used. For the theory, it will be presented by IZZATUL.
Thank you, Ain. Next, I will presenting about theory of paper chromatography. For the paper chromatography, it acts as absorbent, a solid which is capable of attracting and binding the components in a mixture. And the fruit dye is the mixture to be separated that is potted onto the surface of the paper. And the solvent is allowed to seep or flow through the paper by capillary action. There are two components in paper chromatography, which is stationary phase and mobile phase. For the stationary phase, the filter paper holds the sample and absorbs the water molecule. For the mobile phase, the solvent mixture, which is traveled through the stationary phase and capillary action. Next, for the mechanism, there are two mechanisms in paper chromatography. The first one is partition, which separation happens because compounds dissolve differently in the stationary and mobile phase. The stationary phase is a liquid such as water, and the mobile phase is usually a solvent such as alcohol. The compound with higher solubility in the mobile phase move faster and further. For the second mechanism is adsorption, which separation happens because compounds stick differently to the surface of the stationary phase. The stationary phase is a solid such as silica gel, and the mobile phase is a liquid or gas. Compound that absorb strongly to the stationary phase move slower, while weakly absorbed compound move faster. For the sample and apparatus, it will be presented by Wano Atira.
Thank you to IZZATUL for explaining the theory to us just now. Now, I will be explaining about the sample and apparatus that is used in this experiment. The sample that is used in this experiment is liquid food dye. For the apparatus, we will be using sodium chloride, which is table salt, wooden clamp, a beaker, microcapillary tubes, chromatography paper, and also distilled water. We will be continuing with methodology explained by Putri Damia.
Now, we will take a look at the methodology for our chromatography experiment. First, draw a pencil line about 3 cm from the bottom edge of the chromatography paper. Next, mark 4 small X's along the line, spaced about 3 cm apart. Label each X with a different food dye, which is red, blue, green, and yellow. Prepare the solvent by dissolving 2 grams of sodium chloride in 100 milliliters of water. Then, pour about 70 milliliters of this solution into a clean beaker, either 1000 or 2000 milliliters in size. Using a microcapillary, place a small spot of each food dye on its corresponding X. Attach the top of the chromatography paper to a clamp and stand. Carefully lower the paper into a beaker, ensuring that the solvent stays below the pencil line and the paper does not touch the sides of the beaker. Let the solvent rise up the paper until it reaches about halfway. Once that happens, remove the paper and immediately mark the highest point the solvent reached. Let the paper dry for about 5 to 10 minutes. Once dried, draw a line across the marked solvent front. Before we move on to the results, which will be presented by QISTINA, we will now watch a video demonstration of the chromatography experiment to better understand how the process works in practice.
Based on the video we had just watched, these results were achieved from the experiment. Starting with the red dye, a single well-defined spot that moved upward for a moderate distance. It has a stronger attraction to the paper, which has stationary phase. It also has a moderate solubility in the solvent, allowing for steady migration. Next is blue dye. Blue dye traveled the farthest, showed a large spread out spot, a deep blue at the top to a distinct red at the bottom. It has weaker attraction to the paper, allowing it to move quickly and far. It also proves that blue is not a pure dye but a combination. Moving on to the green dye. Green dye separated into two distinct colors, blue at the top and yellow at the bottom. A mixture of two dyes. Blue portion moved farther than the yellow. It proves that green is not a pure dye but a combination. Lastly, yellow dye. Yellow dye forms a bright compact spot that moves a short distance up the paper. It has lower solubility. It also has weaker interaction with the paper, causing it to travel more slowly. Now, let's take a look at the RF value obtained from this experiment. To calculate the RF value for each dye, we can use the formula given, which is RF equals to distance travelled by the solute divides by distance travelled by the solvent. From the calculation, the RF value for red dye is 0.6596. Next, RF value for blue dye is 0.9894. Then, the green dye has the RF value of 0.9468. And lastly, the RF value for yellow dye is 0.5638. From the RF values obtained, we can conclude that higher RF values, blue and green, mean those dyes were more soluble in the solvent and had weaker interactions with the paper. Lower RF values, red and yellow, indicate those dyes were less soluble or had stronger attraction to the stationary phase. That's all from me, and I'll pass the mic to Daniel for the conclusion.
Next, last but not least, is the conclusion. From the conclusion, we can answer the objective of this experiment is to separate food dye using paper chromatography was successfully achieved. As the result that we get from the color green and blue, we can identify it's found to be a mixture, which is, let me separate this. For the green color, it contains two colors, which is a mixture of blue and yellow. Next, blue color is a mixture of two colors, which is blue plus red or dark pink. So how about another two color? We can see that the another two color, which is red and yellow, is found to be a single component because there is no color separation on the paper chromatography. Separation happens because dye move at different speed based on their solubility in the solvent. Clark 2020. Lastly, paper chromatography is a useful method to identify and separate components in food dye. Next, last but not least, is the